Hi, I'm Dr. Arthur Brodsky, the Assistant Director of Scientific Content at the Cancer Research Institute. And today I'm grateful to be joined by Dr. Malay Haldar, a CRI Lloyd J STAR at the University of Pennsylvania. Welcome, Dr. Haldar.
Thank you. It's great to be here.
In cancer, T cells get much of the glory, perhaps because they can target tumor cells directly and eliminate them with precision. But the effectiveness of T cells depends on another group of immune cells that you're exploring called mononuclear phagocytes. These immune cells, which are also known as myeloid cells, or rather are in the group of myeloid cells, they come in diverse types, with some that can promote immune activity and others that can suppress it. And if the latter dominate in the tumor microenvironment, cancers can evade and escape immune responses. So I was hoping you could share with us a little about what are the different types of these myeloid cells, and what do we know about their roles that they normally play, as well as in cancer?
So as you mentioned before, myeloid cells and lymphoid cells are different types of white blood cells that participate in immune responses. They come from different precursors in the bone marrow and have distinct functions. And there are different types of myeloid cells, and the ones we focus mostly in our lab are known as antigen-presenting cells. I think when you're thinking in terms of the roles of these different cell types in cancer, a good analogy is to think about the immune system as a well-organized military or the army. Lymphocytes, much like your T cells and B cells, are the soldiers of the army. They are primary cells responsible for killing the tumors, and therefore, as you pointed out, they have garnered a lot of attention for cancer immunotherapy. However, they need instructions and training so that they can distinguish a cancer cell from normal cells in the body. And this instruction is given by antigen-presenting cells. And so you can think of these antigen-presenting cells much like the generals in the military or the army. They collect information from different sources to distinguish a cancer threat from normal tissue in the body. Then they convey this information to the T cells, telling them exactly what to look for, how to look for these cancer cells, and what strategies they might use to neutralize these cells. And armed with this information, T cells then go to tumors to eradicate them. And so perhaps with this analogy, it's somewhat easier to appreciate that for tumors to avoid immune responses, a good strategy would be to convince these antigen-presenting myeloid cells that tumors are not a threat. And the tumors can do this by various different ways. They can start recruiting more of immunosuppressive macrophages, which are a type of myeloid cells that can shut down the function of anti-tumor T cells, or tumors can inhibit functions of another type of antigen-presenting myeloid cells known as dendritic cells. So apart from these macrophages and dendritic cell, another type of myeloid cell that are important to us is known as monocytes. And monocytes can actually differentiate into macrophages or dendritic cells, and tumors often interfere with this differentiation process for their own benefit. And so besides monocytes, macrophages, and DCs that I just mentioned, there are also other types of myeloid cells like neutrophils, basophils, but they're relatively rare inside the tumors, and we know much less about their roles in cancer immunity.
So I just want to clear up something, I guess, for our listeners. You mentioned antigen-presenting cells, and just wanted to clear up the antigens are those they're the ones that tell the T cells what the threat looks like, in this case, cancer. And so the antigen is just that marker that is the specific what molecule to actually go after for the T cells. So as a CRI STAR, you're exploring how tumors and these myeloid cells communicate. So what are some of the important outstanding questions regarding myeloid cells and their impact on cancer immunity? And how might your work help us overcome some of the current limitations of cancer immunotherapy and improve care for patients?
Right. So despite all the recent successes and excitement with T-cell focused immunotherapy, there are several major hurdles in the field. One is, as I mentioned before, the body does not recognize tumors as a threat, and therefore does not generate enough of anti-tumor T cells. And the other bottleneck is, even if you have generated anti-tumor T cells either naturally or through treatments, they are often shut down in the tumor microenvironment by these suppressive myeloid cells. So what scientists have realized in recent years is that tumors are constantly trying to inhibit the so-called good myeloid cells, such as the dendritic cells, while at the same time promoting bad myeloid cells, such as the suppressive macrophages. What we have also realized is that different tumors employ different strategies to achieve this through communication between cancer and myeloid cells. So my group is trying to understand the molecular language of this communication with the overall goal of using this information to alter myeloid cell behavior and their distribution in tumors for immunotherapy. And so one example of the this type of work is a recent publication from our group showing that some tumors produce large amounts of retinoic acid, which causes monocytes to generate more of that immunosuppressive macrophages I mentioned before. And importantly, if we inhibit retinoic acid activity in these tumors, we can now generate fewer macrophages and more dendritic cell. And the end result of this is that we generate a stronger, more robust anti-tumor immune responses. So we are currently building on this discovery by developing novel inhibitors of retinoic acid production and signaling, with the goal of taking these to clinical trials in the near future. We have also recently discovered other pathways that are distinct from the retinoic acid-based pathway I just mentioned, and we're trying to understand how these other pathways also regulate anti-tumor immune responses through myeloid cells. And the overarching goal always is to find therapeutic vulnerabilities that we can exploit for new treatment approaches.
Interesting. So essentially, it sounds like you're kind of trying to disrupt that bad feedback cycle that gives rise to the myeloid cells that shut down the T cells. And then, you know, I guess if you could disrupt that, then the T cells, not only might they be able to do their job better naturally without extra help, but then presumably also the checkpoint immunotherapies and others that target the T cells, then they might work better too, correct?
That's right. That's absolutely right. Yep.
And so as you're going about answering these challenges, or answering these questions, what are some of the biggest challenges as far as going after as far as learning more about myeloid biology? And are there any technological challenges that you're addressing at the same time in order to answer these questions?
Right. So the cells, the myeloid cell specific that I just mentioned before, comes in various different functional types. Furthermore, different types of tumors would often contain different subtypes of these cells. So a major challenge currently is to understand which tumors harbor what types of these cells and how might these be affecting the anti-tumor immune responses. The other challenge that I also mentioned before is that we have very limited understanding of this crosstalk between cancer and myeloid cells, by which the tumors really convert some of these myeloid cells from a foe to an ally. As in many cases, we know that intratumoral myeloid cells can end up helping the cancer grow and spread. So to address some of these challenges, which is mostly related to diversity and heterogeneity of these myeloid cells, we are employing now cutting-edge single cell technologies using both mouse models as well as human tumors.
And you mentioned the single cell technologies. I would think this is especially important, you know, given you've been talking about the kind of diversity and complexity of these myeloid cells. It's not kind of the current state of understanding. It's not quite clear necessarily what makes the progenitors that give rise to all the other subsets, what kind of causes this. So hopefully, you know, you'll be able to make some progress there. So zooming out a little bit, what do you hope to accomplish over the next 5 years as a CRI STAR, and how do you hope that your work will impact the field?
So in the next 5 years, we really hope to discover additional pathways, much like the retinoic acid-based pathway I just described before, that underlie this communication between cancer and myeloid cells. Another major hope is that we will be able to transition some of our basic discoveries into clinical trials. We're actually already engaged in one such study where we are examining the safety and efficacy of a new compound. It's a first-in-class compound that we have jointly developed with NIH to target the retinoic acid pathway, specifically in liver cancer to begin with. And our hope is that we will be able to start a phase 1 trial very soon once we have completed these safety studies. So generally speaking, the overall hope is that our work will impact both the basic understanding of tumor immunity as well as the landscape of cancer therapy.
That's great to hear. I mean, obviously the basic science is important and the discoveries are exciting, but at the end of the day, it's all about helping people.
Right.
So now, you know, I think the public, there's not a lot of understanding necessarily about how scientific grants work and how different programs supply funding. So I was hoping, could you explain why this CRI STAR funding is so important for your research? And specifically, what will this support enable you to pursue that you might not have been able to otherwise?
Right. So a key component of this award is that it is really not tied to a very specific project. So what happens normally in a funded project is that we end up discovering new and completely unanticipated things during the course of the study, right? And so, but it's very difficult to pursue this exciting but new lines of investigation because generally speaking, most fundings have a very narrow focus. So if you wanted to do that, if you wanted to pursue these new lines of investigation, you will have to write a new grant, wait for it to be reviewed, get the funding, and then start. In contrast, the CRI funding is very different as it does not really restrict us to a very specific line of investigation. It's broadly encompassing tumor immunotherapy. So this really allows us to be nimble and expedite and actually enable studies that would be otherwise very difficult to pursue. In summary, this award really enhances both the type and the pace of work in our lab.
I think you bring up a great point that science is not something where we necessarily know, not the destination, but the journey is it's mysterious. That's why we have to kind of go into the unknown to answer those questions. So thank you very much, Dr. Haldar, for taking the time to speak with me today, and great luck in your work. I can't wait to follow your progress.
Thanks for the interview and thanks for your support. It's really a pleasure.